Our next speaker is Jun Tao. Jun is a director of in vitro pharmacology at iXCells. Her team focuses on supporting rare disease patients through antisense oligos, siRNA, and small molecule screening, contributing significantly to advanced N-of-1 therapies with multiple patient foundations here. With a PhD from New York University and postdoc training at Harvard University, Jun brings academia excellence and over 11 years of experience from roles at Amgen and WuXi AppTec in drug discovery and CRO services. Jun, the stage is yours.
All right. Good afternoon, everyone, and welcome to iXCells. So my presentation today has 3 parts. First, I will give a very brief overview of our company. And then the second part is the disease-relevant cell models. And number 3 is the drug screening platforms. So first, I would like to give you a very brief overview of our company. iXCells Technologies was founded in 2014, headquartered in San Diego, California. And over the past 10 years, the company has undergone remarkable transformation by recruiting experienced commercial staff and executive members, as well as the talented scientists, building the infrastructure and expanding our pipeline, product pipelines, and also CRO services. So today, as you all know, we are celebrating the grand opening of this beautiful new facility, which is definitely a milestone for us moving forward to the next phase of growth. So our expertise is to provide innovative solutions for preclinical drug discovery. And we offer both products, cellular products, as well as preclinical CRO services. So I will share some details with you in the later part of my presentation. So moving to the second part, which is the disease-relevant cell models. So this slide is a summary to show you our business model. We offer 2 major categories of cellular products. One is primary cells. The other is iPSC and iPSC-derived derivatives. So in addition to the ready-to-use cryopreserved cells, we can also perform customized primary cell isolation, as well as iPS reprogramming. So we can utilize the cutting-edge technology CRISPR to manipulate the iPS, either by inducing a mutation or by correcting a mutation. So the iPS cells can be targeted, differentiated into different cell types, including different types of neurons and other cell types like hepatocytes. And those cells can serve as cellular models for different stages of studies in drug discovery pipeline, including target identification, drug screening, functional assays, as well as toxicity assessment. Primary cells are valuable tools for preclinical studies. So we have a diverse collection of primary cells, including multiple cell types as listed here, and epithelial cells, endothelial cells, muscle and neurons, etc., isolated from a variety of tissues as listed here. And those cells can be from animals, different species, rat, mouse, etc. And also we have a large collection of human adult donor primary cells, including individual, healthy individuals, as well as disease patients. So, and we can also conduct multiple cell isolation, primary cells, from the same donor, which is a very powerful approach to study different cells from the same donor. And in terms of delivery, we have different delivery methods and platforms. The primary cells can be shipped to our customers either in the frozen vials or in the fresh culture in the culture dishes, just depending on our customers' needs. We also have a very stringent QC process to ensure the quality of our primary products. Induced pluripotent stem cell, or so-called iPSC, is another expertise areas at iXCells. So we helped many pharmaceutical companies as well as rare disease patient families to generate iPS lines from the either PBMCs or skin fibroblasts. So we have a collection of both healthy donors iPSC lines as well as disease iPS lines. And as I mentioned earlier, we are capable to use CRISPR technology to change the manipulate gene expression in the iPS lines by either introducing a mutant or correcting a mutant. All the iPS lines generated in-house, we perform a comprehensive characterization, including cell variability test and the alkaline phosphate staining and iPS marker staining, as well as cryotyping. And again, all the cell lines has to pass the our QC, make sure it's mycoplasma free and pathogen free. So all the iPS cells can be differentiated into multiple cell types, as you all know, which is a revolutionary approach. So since our focus is neuronal diseases, we are able to produce multiple types of neurons, and those are our catalog products, including a cortical neuron, motor neuron, and GABA neuron, and dopaminergic neurons. And all of these different types of neurons have been used in our in-house CRO services, as well as cellular products that you can purchase. So we can also differentiate the different other cell types from the iPS, including muscle cells and hepatocytes. So our team is working really hard to keep this list going longer and longer. Moving to the third part, as I mentioned, so we are not only offering cellular products, we are also providing preclinical CRO services. So drug screening platform is one of our services. And one of our focus right now is to develop personalized therapy to treat rare diseases. As you can see here, I listed several cellular models, and I use neuronal diseases as an example, which we can utilize either both immortalized cell lines or iPS-derived neurons, as well as primary cells. So later in my presentation, I'm going to show you a summary table to compare the differences and our recommendations for these different cellular models. And those cellular models in our hands empowered us to work on multiple drug modalities, including, but definitely not limited to, RNA-based therapies, including ASO, antisense oligo, siRNA, and we can also work on virus-based gene therapies to deliver mRNA, CRISPR, and base editor. We are able to evaluate small molecules as well, either by screening large commercial libraries or a smaller customized collections. And the last item is so-called drug repurposing, which means we try to uncover a new indication for FDA-approved drugs to use for a new disease. So we have a standard protocol, a procedure for drug screening. Here, I'm just showing you an example of this flow chart is a typical ASO screening performed in patient cells. So the very first stage is always a discussion with our customers. And most of the time, those are private foundations and the rare disease families or research labs. So we try to understand the biology and understand the needs of our customers. And the next step would be to generate patient-derived iPS cells, and then differentiate those cells into the disease-relevant cell type. And then we need to design the ASO, synthesize the ASO, and perform assay development, followed by high-throughput screening. Of course, the hits we identified needs to be conformed and characterized. So to help the rare disease patients, and especially the young kids, we understand we are not only fighting with the diseases, and we are also most of the time fighting against the time. So one of the most common questions we always receive from the patient's parents is, how long does it take to develop an ASO treatment? So here I'm showing you a timeline estimation. And this estimate is based on the development of Milasen. And you heard a story from Julia earlier this morning, which is Mila's mom. And you also heard the presentation from Dr. Tim Yu, who led the efforts to develop this personalized ASO treatment. So as shown here, it is possible within 10 months to accomplish all the stages listed here. And as you may notice, some of the activities happened at the same time in parallel. For example, generating patient's iPS cells and also produce the iPS-derived neuron takes up to 6 months. But while this effort is ongoing, we can start ASO design and synthesize. While we are waiting for the ASO to be synthesized, we can start the assay development in immortal cell lines. And also we can perform the primary screening in the cell lines. So when we identify the good hits from the primary screening, hopefully the iPS cell at that point is ready. Then we can further conform, validate, and characterize, optimize those hits in the primary cell lines, in the iPS-derived lines. So we have been using this strategy in multiple projects, using this in parallel, different arms in parallel strategy, which worked really well. It dramatically shortened the timeline for the ASO drug development process. But I have to say, each case is unique. So the timeline may vary for each individual patient and each individual case. And another common question we always receive from the parents is, which cell system to be used? So I'm showing you here is a comparison table listing 4 different cellular models. So on the very left side is the immortalized cell line. So the advantage of using the immortalized cell line is it's very easy to access and very low cost and almost unlimited sources. Of course, we all know the disease relevance, the score is a little bit lower compared with iPS-derived cells, but I think it's still a very useful tool or cellular system to be used especially at the early stage of the ASO drug development process. For example, for assay development or primary screening. And on the right-hand side, for the iPS neurons, of course, nobody would argue. The iNeuron from the patient is the most biological relevant system. However, we all understand it takes longer time, so the cost will be higher. And something in the middle would be a wild type or a normal iPS cells, iPS neurons. And since we have a collection of wild type iPS, different iPS lines and different types of neurons, so that will save us some time and also some cost. And in some cases, we can utilize the wild type line, iNeurons to perform the screening. So moving forward, this is a very simplified chart to summarize how ASO works. So we can utilize this very small oligo, a short oligo nucleotide antisense to either increase or decrease the target gene expression. So for downregulation, two well-known mechanisms are RNase H mediated mRNA degradation, and the other one is so-called steric blocking to stop translation. And for upregulation, one mechanism is to inhibit naturally existing microRNAs or antisense, or a so-called splicing modulation, either by exon skipping or exon inclusion. We have been working on multiple ASO screening projects. So I'm going to show you some examples now, which utilize different mechanism of action for the ASOs. So this project, we collaborated with PCH1 Foundation. And you heard the story from Michelle earlier this morning. So the goal here is try to identify knockdown ASO in immortalized cell lines. So we utilized a cell line called, which is called SHSY5Y cell, is a neuroblastoma cell line. So we screened a relatively small collection of ASO, only 30 ASOs, but we are very, very lucky to identify 2 of them give us inhibition effect on the target gene. And then we moved on to the next stage, hit confirmation. As you can see that both of these hits give us very nice dose-dependent gene knockdown effect. So the third stage will be hit validation in iNeuron, which is ongoing just next door in our lab. So hopefully in early next week, we will get the results. Case study number 2, collaborated with India Accelerator, as well as Boston Children's Hospital and Crayon. So the goal here for this project is to identify allele-specific knockdown ASO in patient iPSC-derived neurons. As you can tell here, we are measuring or monitoring both the expression of the wild-type allele represented by the blue dots, or the mutant allele expression represented by the red dots. And higher concentration of ASO is the darker color. And we have 3 concentrations for each ASO. And for this particular project, we screened a very large ASO library, about, I would say, over 800 ASOs total, because it's not easy to identify an allele-specific ASO, knockdown ASO. But as you can tell, at least in this orange box I highlighted here, these 3 ASOs give us dose-dependent downregulation of the mutant allele, but has a very minimal or small effect on the wild-type allele. Case study number 3. So this is a collaboration between iXCells and the Cradle-Chat Foundation, as well as MEHI. And the goal here is to restore a target expression by targeting microRNA. There are microRNAs naturally exist, which can inhibit gene expression. So the first stage for this project, we evaluated 11 microRNAs in an immortalized cell line. And we are very lucky to identify 3 microRNAs showed inhibitory effect on the target gene. Stage 2, we wanted to select which cell type, which cell system we are going to use for ASO screening. And we evaluated the 3 identified microRNAs, both in the immortalized cell line, as well as 2 NPC neuron progenitor cell lines. And we found that neuron progenitor cell has similar or even higher levels, endogenous levels of the microRNAs. So now we have the candidate microRNA, and we have the identified cell model. the next stage would be to perform ASO screening, try to identify the ASOs which can inhibit those naturally exist microRNA. And we are currently in the discussion phase for the ASO screening. So hopefully we can initiate the screening very soon. Okay, so those three examples are just some cases among all the ASO projects we have been doing so far. And as you can tell, we are collaborating with multiple patient families, private foundations, as well as research labs. So, and we worked on different diseases and using different design strategies and so on. So definitely this is a very educational process for our team as well. We are learning a lot, gaining experience. So hopefully we can serve our patients better. And here is a list of small molecule projects for rare disease. So for rare diseases, we collaborated with Rarebase and to evaluate small molecules. And some of these projects are so-called drug repurposing, trying to identify new indications for the FDA-approved drugs. So we work closely and collaboratively with all our partners within the rare disease community. As you can tell from here that the ecosystem is built and with the contributions from biofarms, from research labs, as well as patient families and foundations. So I think we need to work together really hard, support each other, try our best to save and help those lovely kids. Finally, I want to say thank you to all our collaborators. The list here is definitely not exclusive. Those are just some organizations whose projects were referenced in my presentation today. I also want to thank the whole iXCells team. So today's presentation is definitely a joint effort, a team effort. And thank you very much for your all.